I am Madhumita and I am here to share about EMG. So what is EMG? EMG means electromyograph. Electro means electrical, myo means muscle, graph means pictorial representation. So electromyograph is an instrument which is used to record the electrical activities of the muscle. From that we can determine whether the muscle is contracting or not. So muscle contraction is caused by devolution of the muscle fibers. So the devolution refers to the increase in the negative charge of the cell compared to the outside of the cell. So there are two types of neuron, that is sensory neuron and motor neuron. Sensory neuron, this neuron sends a signal from different body parts to the brain, and motor neuron, it sends signals from the brain to the other body parts. Next, we are going to say about electrodes used for EMG. There are two types of electrodes, surface electrodes and needle electrodes. Surface electrodes. These electrodes are placed in contact with the skin so that we can obtain the bioelectric potential. And there are three electrodes placed on the surface, that is positive, negative, and ground electrodes. Ground electrodes is placed away from the remaining electrodes. Next, needle electrodes. These are inserted inside the tissues so that we can measure the electrical activity of the muscle. Next, we will look into the block diagram of EMG recording system. The electrodes which is placed on the body parts of the skin is given as input to the differential amplifier. Then this output is given to the power amplifier and it is sent to the speaker, or it can be given to the tape recorder and which is used for recording, or it can be given to the storage oscilloscope which displays the waveform. Next, we will look into each of them in detail. This electrodes, EMG potential are taken from the tissue by using these electrodes. Next, the EMG bandwidth is large. Next, differential amplifier. This is nothing but the high gain amplifier. Frequency ranges of this differential amplifier is 10 Hz to 10 kHz, and CMMR, that is common mode rejection ratio, is 80 to 100 decibel. Power amplifier. Power amplifier is used to amplify the signal. Next, oscilloscope. It is used to display the EMG waveform. From that, we can observe the abnormality of the nerve and muscle cells. Next is tape recorder. The signal can be recorded using this tape recorder. Next, we will look into the nerve conduction velocity measurements. It is used to find the location and type of the nerve damages. The nerve function is examined directly at the various stages of the nerve by stimulating the nerve with a brief electric shock. This electric shock, a pulse duration has to range from 0.2 to 0.5 milliseconds. From that, we can measure the latencies. Latency is nothing but the elapsed time between stimulating impulse and muscle action potential. From this, we can calculate the conduction velocity in the nerve. In the waveform, the distance between the stimulus and the muscle action potential is the distal latency. Next, the positive and negative cycle is the total duration. The electrodes are placed in two different parts of the parts on the body. That is, one in distal simulation and another is a proximal simulation. From that, two different waveforms are obtained, and the result is finalized. Next, we look into the advantages of the EMG. It is used to diagnose neurogenic or myogenic diseases, and we can find symptoms of numbness, muscle weakness, paralysis, and it is used to detect the muscle fatigues at early stage. Next is disadvantages of EMG. As we are using needle electrodes, it is quite painful and it is less accurate due to the disturbance from surroundings, and setup is fairly time consuming. Next, the applications of EMG. That is, it is used in electrophysiological testing, which is a branch of physiology that studies electrical properties of biological cells. Next is clinical neurophysiology and neurology, which is related to nerves, and psychiatry, which is related to mental disorder. Thank you.